this is part 2 of the recorded lectures on cell division. We left off on this slide. We didn't get a chance to finish it. So basically, this is just an overview of the phases of the cell cycle. So you have interphase, and during interphase, the cell grows and the DNA is replicated. Primarily, these are the two events that occur during interphase. Interphase is going to be divided into G1, S, and G2, which we'll discuss a little bit later. And then there's the mitotic phase, and during the mitotic phase, the two sets of DNA, since the DNA has replicated, we now have two sets of DNA. So the replicated DNA and the replicated organelles that are in the cytoplasm, all of these are separated, moved to each side of the parent cell, and then that cell divides down the middle. So you have two daughter cells that are clones of the parent cell. And this is an illustration of the cell cycle. Says cell cycle start, so we'll start right here. This is interphase, or okay, what you have to remember is during all of these interphase stages of the cell cycle, they're showing you the chromosomes, but the chromosomes are not condensed. They don't actually condense to where you can see them until prophase. If you were looking at interphase of this cell that I have, where I have the arrow, if you were looking at that under a microscope, you'd have the cell, and then I'm not drawing it in yellow, but that nucleus in there would be colored all in a solid color, because the DNA is in the form called chromatin, and it is spread out so much that it just looks like a solid color all throughout the nucleus. But anyway, it's showing you here that there are 4 total chromosomes at the beginning of the cell cycle in interphase, or G1. And then in the S phase, the chromosomes are all replicated or duplicated. So you can see that you started out with 4 chromosomes. You still have 4 chromosomes, but they are 4 duplicated chromosomes. Each chromosome has made a copy of itself. And we will discuss that a little more later on as well. And then we have a phase called G2. That is a second growth phase where different things occur, but probably the most important is that these fibers that you see here, down here in metaphase, they're called spindle fibers or spindle microtubules. Those are produced during that G2 phase. So we've got G1, S, and G2. These are the phases of interphase. And then we have the phases of mitosis, beginning with prophase, which is where the nucleus disappears and the chromosomes condense, and you can see them. They're not in the form of chromatin anymore. They are condensed into these structures that look like X's, because they're actually 2 sister chromatids attached at the center. And then in metaphase, the chromosomes line up at the metaphase plate or the equator of the cell. In anaphase, the sister chromatids of each chromosome are pulled apart, and so they are pulled to opposite ends of the cell. And then in telophase, all of these have arrived at the ends of the cell, and you can see the cells start to pinch in at the middle if it's an animal cell. And that shows you that it's dividing. And when it does divide, that's called cytokinesis. You're going to have 2 daughter cells identical to the parent. Here's your parent cell. These are your daughter cells. Now, we know that some cells in the human body, once they are produced, they do not divide anymore, and like nerve cells, for example. So those cells will then go into a G0 stage, which means that they won't continue going, excuse me, that will be the end of the cell cycle for them. They will not necessarily die. They can go on and live for years and years and years, but they will not divide anymore if they're in the G0 stage. So, beginning with the stages of interphase, excuse me, we know that there are three stages called G1, S, and G2. I think of the G as growth and the S as synthesis. They call it the first gap phase, but I just always remember it. G stands for growth because that's really what's happening. So the G1 phase, the first gap phase or growth phase, is when a lot of things are going on biochemically. The cell is accumulating the building blocks of chromosomal DNA and the associated proteins, as well as accumulating sufficient energy reserves to complete the task of replicating each chromosome in the nucleus. So it is preparing to replicate the DNA. The building blocks of chromosomal DNA are going to be DNA is composed of nucleotides. Remember, those are the building blocks of DNA, the A, T, C, and G. All of those nucleotides plus the DNA is wound around histone proteins, so more histone proteins need to be produced, and various molecules like that. So the S phase is when I always think of it as the synthesis phase. That is when the DNA actually replicates. DNA replication occurs, and we have a chapter where we learn how that process occurs. But that is when we have one single chromosome replicating itself, and now we have one chromosome that is composed of two sister chromatids that are attached at a region called the centromeric region. The centrosome, which consists of two centrioles, is also duplicated, and the two centrosomes are going to give rise to the spindle, the mitotic spindle, and that is what I showed you here. All of the fibers, they kind of look like a football shape. That is the spindle. That is the spindle fibers attached to the sister chromatids of each chromosome. And what they're doing is lining them up at the metaphase plate, and then those spindle fibers are going to shorten and pull the sister chromatids apart. Okay, now let's see. And this just says that there are two centrioles in animal cells at the center of each centrosome. So the centrioles help organize cell division, but they're only present in animal cells. They're not present in plants and fungi. But centrosomes are, but only animal centrosomes contain centrioles. In the G2 phase, or the second gap phase, or second growth phase, whatever works for you, is when not only, because the DNA already was replicated, but now some of the cell organelles are going to be replicated or duplicated. And maybe the cell grows a little bit more during that stage as well. In the mitotic phase, known as the M phase, the first part of the mitotic phase is called karyokinesis, or nuclear division. And the second part is cytokinesis, or the division of the cytoplasm, the actual splitting of the cell into two daughter cells. Okay, so here's another diagram. Here's our parent cell, and these structures here are your centrosomes. And this is an animal cell, so the centrosomes contain centrioles, but they don't always contain centrioles. Um, so during prophase, as you can see in the nucleus, during prophase, at the beginning of prophase, the DNA is still spread out. It's still in a form we call chromatin, but it is condensing into chromosomes. And then we have a stage called prometaphase. It's just in between prophase and metaphase. That is when the spindle fibers have formed. You can see them, and they have connected to each of the pairs of sister chromatids, each of the chromosomes, each of the duplicated chromosomes. And then those spindle fibers help to line the chromosomes up at the metaphase plate. The spindle fibers shorten and pull the sister chromatids to the opposite ends of the cell. And now in telophase, the nuclear envelope reforms around the two new sets of chromosomes. And you're going to see the centrosomes here and here, they're going to disappear. And then cytokinesis will occur where a cleavage furrow forms in animal cells. It's called a cleavage furrow. And then the cells just pinch into, and now you have two daughter cells that are clones of the parent cell. This is your parent cell. In prophase, there's a lot of information here, but let's just see what's the most important. The nuclear envelope begins to disappear. It begins to dissolve or dissociate. Okay. The nucleolus disappears. The centrosomes begin to move toward opposite poles of the cell. Prophase is the stage that has the most events occur during prophase. If you can learn prophase, the rest of the stages are easy because when you get to telophase, everything in telophase is opposite of prophase. So the hardest stage to learn is definitely prophase. So you spend a little time learning prophase and the rest will be easy. The microtubules that will form the mitotic spindle extend between the centrosomes and they push them farther apart, and the microtubule fibers lengthen. And the sister chromatids begin to coil more tightly or condense into chromosomes. In other words, the DNA was chromatin, which is very spread out, and then it condenses into chromosomes during prophase. So visible chromosomes that you can see under a microscope. In prometaphase, the spindle microtubules attach to the kinetochores of the sister chromatids. So the microtubules will bind to the kinetochores of the sister chromatids. And the kinetochores, okay, this shows you. All right, so this is a pair of sister chromatids. It's only considered as one chromosome, but it's a duplicated chromosome that contains two sister chromatids. The centromeric region is the center where the sister chromatids are attached, and these structures are called kinetochores. Okay, these structures here and here are called kinetochores. This is where the microtubules are going to attach to the sister chromatids and line them up. So in metaphase, the chromosomes are going to be lined up at the metaphase plate. And then in anaphase, the cohesin proteins, which had been holding the sister chromatids together, degrade, And now each sister chromatid, which is called a daughter chromosome at this point, is pulled toward the